Good morning, good afternoon, good evening to all our participants around the world, US, Canada, everyone. Welcome to our spring Going Organic webinar series. My name is Dil Tavaraja. I'm a professor at the Clemson University, oversee the Pulse Quality and Nutritional Breeding Program at the Plant and Environmental Sciences, Clemson University. Our Going Organic project and the webinar series is funded by the National Institute of Food and Agriculture, OREI program. Today is our 2025 Spring Going Organic last webinar. We have been conducting this since the fall, and we had a range of different researchers around the world, especially in the US, growers as well as the researchers and the breeders. We brought many people and discussed about how we can improve our organic agriculture and share their experience in organic plant breeding, nutrition, marketing, processing, and policies. Today, we are wrapping our spring webinar series with 3 of my doctoral students in my program. Before I introduce the speakers, I wanted to thank Mr. Tristan Lawrence, who is the program manager and take care of everything and organizing, advertising regarding to the organic series. And also, I like to give a little bit of housekeeping. Please introduce yourself, name, institute, and also when you have questions, send us the question through Q&A, and we will try to answer all the questions because we have 3 speakers today, and they are going to speak only 15 minutes. First, we will listen to all 3 and then take the questions at the end. So our first speaker is Ms. Sonia Salaria. Sonia is from Punjab, India. She has a BS and master's degree from Punjab Agriculture University. She joined my program in 2021. She is a plant breeding and genetics candidate right now, and she is planning to graduate in August 2025. Our second speaker is Amod Madurapperumage. Amod is originally from Sri Lanka. Amod is a chemist. He has a master's, not the master's. He has a BS degree from Institute of Chemistry, Sri Lanka. And he also joined my program in 2021. And Amod is working on high throughput technology for plant breeding. And he is planning to graduate in next year. And our last speaker is Mr. Mark Dempsey. Mark has more than 10 years of experience in organic agriculture. He has a master's degree from University of Miami of Ohio, and he will be discussing on lentil diseases and how this new crop adapted to South Carolina. With that, I would like Sonia to start the talk. Thank you, Sonia, Mark, and Amod.
Yeah, thank you so much, Dr. Dil. Greetings to all who are joined today. I'm here to present a part of my research, which is about breeding for protein-rich organic lentils. So briefly introducing lentil crop. So it was originated in the Middle East approximately 8,000 years ago, and from there it spread across the different countries. Botanically, lentils belong to the Fabaceae family or Leguminosae family, and the wild progenitor close to it is Lens orientalis. Lentil crop has 4 gene pools, and the 4 gene pools based on their ability to interbreed with each other. So there are 4 gene pools. The primary gene pool has the species which can closely and easily intermate with each other. And as we move from primary to quaternary gene pools, the mating becomes difficult. So lentils, they are the annual plants which have erect to semi-erect stature, and they have indeterminate growth habits. The plant color for lentil ranges from pale green color to dark green color, and the flower in lentils are complete, which means they have both male and female parts, and which helps in facilitating self-pollination. So the 2 types of lentil flower colors are white and blue. Also, based on the seed size of lentil, there are two types, macrosperma and microsperma. Macrosperma are the last seeded one, which has a seed diameter of 6 to 9 millimeters, and the small seeded ones are microsperma, which has diameter of 2 to 4 millimeters. And nutritionally, these are rich in proteins, which is approximately 25% in them, and they have a good amount of complex carbohydrates, and also they have no fats in the seeds. So lentils can be used in numerous ways. So from the store-bought lentils, you can make the curries, soups, salads, and the fritters as indicated in the pictures here. And they can also be bought as a lentil market products. So you can buy them from market as a pasta in terms of and chips and different crackers and soups. So you can use them in numerous ways. So coming to the production of lentils, so lentil is being produced in 55 plus countries, and here are the top 5 countries according to the data recorded in 2023. So you can see here Australia is the top lentil producing country, while US comes at the rank 5th. Now coming to the production of lentils in USA, so the lentils are mostly concentrated in the 4 or 5 states you can see here. So they are highlighted here in the green colors, and the color intensity of the shows that how much the lentil is produced in each state. So Montana is the top lentil producing state. And if you see the southeastern region, it has no state which produce lentils, which means the lentils are not being adapted in here. So it is a great opportunity to breed lentils for their adaptation in southeast. And since the production of lentils has increased so much from the 2023 compared to 2024, which is probably due to increase in the consumer demand and in the use of the lentils by the consumers. So which means we need to improve the production to meet the demand. So this opportunity is a good thing to pursue. So coming to why should we breed for lentils, other than their adaptation to southeast, so they are a good plant protein source, and they can be used as a good source of vegetarian plant diet. So it is a good candidate crop for plant-based protein industry. And also lentils are the sustainable crops. They can be grown only as a rain-fed crop, so they use very less amount of water. They can save the natural water resources. And other than that, it is a legume crop, which is a cool season. It can fit into the winter cultivation because of the shorter duration, so it can act as a new cash crop for the southeast growers. And lastly, they are the nitrogen fixers. So lentil has a root nodules which contain rhizobium bacteria which fix atmospheric nitrogen, and which means we do not need to apply nitrogen fertilizers for them. So this is also an environmentally sustainable crop. So since it is a protein-rich crop, so most of the studies were performed for protein improvement, and many studies were classical. So there were a very few studies which were conducted by using genomic approaches. So here in this table, you can see only 3 studies I've been able to find so far. And the protein range in the classical studies was reported to be 30%. So it means it has a huge potential to improve on protein concentration and protein quality in overall. Other point of lentil protein is it is an imbalanced protein. It has a limiting concentration of essential and sulfur-containing amino acids. So in this picture, you can see the different pathways. So it is a very complex pathway for the synthesis of different amino acids. So here the essential amino acids are indicated in green boxes, and the non-essential amino acids are indicated in the purple boxes. So because of the complexity of different pathways, it is difficult to breed for all of the amino acids at once. So therefore, we can focus on a few of the amino acids. So in our study, we are focusing on protein quality, and it includes amino acids. So the 2 amino acids we are focusing are sulfur-containing amino acids, which are cysteine and methionine. So to conduct our study, we assembled a lentil association mapping panel. So it has 3 sub-panels, which are Global Association Mapping Panel and Heat Tolerance Population. These two are sourced from ICARDA, and one is a lentil single pan selection, which is sourced from USDA. So in together, it contains 446 accessions. So these accessions have their origin from 55 plus different countries. So here in this map, you can see the different countries from where they have been sourced. And in the pie graph, you can see the proportion of panel which comes from each continent. So our panel mostly belongs to the Asia. So by using this panel, we conducted a greenhouse experiment. So we conducted this experiment for 2 years. We used a total 446 accessions in this, and we laid out it in completely randomized design with a varying number of replications in each year. We also used a check, CDC Maxim, and we collected data for protein quality traits. The 3 traits we used were protein concentration, sulfur-containing amino acids, and protein digestibility. So here I will be just showing a brief results about the greenhouse experiment. So in the first 3 distribution graphs, so the first trait is protein concentration, second is sulfur-containing amino acids, and the last one is protein digestibility. So in these distribution graphs, the yellow color line shows the overall mean for that particular population for a trait, and the purple line indicates the value of mean value for the check. So for protein concentration and protein digestibility, the overall mean value for the population was less than the check, but for sulfur-containing amino acids, it was similar to what we saw in check. But if we see the these plots, so in these plots, you can see the different outliers. So in each trait, the number of outliers are very less, which are indicated by the red dots. And even if in the distribution graph, we see that the overall mean is less, but we can see that all the traits have a fair number of accessions, which have higher mean value than the check. So we have a good number of accessions to select from for our breeding program. So by using the data, we conducted analysis of variance for all these traits, and the model we used here include the genotype, year, replication as a variable. So here we considered genotype as a fixed factor while other factors were considered as random. So in here we found significant genotypic effects for all 3 traits, while the other effects were non-significant. And for the interaction for genotype and year, it was found significant only for sulfur-containing amino acids. Along with this, we also conducted the correlation analysis, and we found the positive correlation for all the traits, and the correlation values were very high for all the traits, which means that we can target the improvement of all 3 traits at once, which is good. Then we conducted a principal component analysis. So in here we have seen the first principal component was explaining 75% of variation, and the second one was explaining 16%. So in combined together, they were explaining more than 90% of the variation in the phenotypic data. And in the PCA bioplot, you can see the different variables as vectors indicated in green color. So the sulfur-containing amino acids and proteins were contributing positively to the principal component 2 and negatively to principal component 1. And the protein digestibility was contributing negative to both the principal components, which means that with the increase in the sulfur-containing amino acid and proteins, the principal component 1 will be decreased and the principal component 2 will be increased, and the similar things for the protein digestibility. Then we also conducted the cluster analysis for all these 3 traits, and we found 4 clusters. And this is a hierarchical clustering we used. And the cluster number 2 was appropriate with the accessions, which has high values of all these 3 traits. And in here, I have highlighted a few of the genotypes which are common for these traits. So the genotypes highlighted in green, they were having high concentration of protein and sulfur-containing amino acids. And these accessions which are highlighted in the blue boxes, they are having high concentrations of sulfur-containing amino acids and high concentration of protein digestibility. So it is good to have the accessions which have high values of these traits, or these 2 or 3 traits, so that we can improve them in together. Then we also conducted the field experiment for 3 years. So for the first year, we conducted experiment at 1 location, but for the second and third year, we conducted the experiment at 2 locations. So in the first year, in the first 2 years, we used the LSP only, and then we did the seed increase, and we used the complete whole panel in the year 2024. And we laid out all these experiments in alpha lattice design in 2 replications, and we performed data collection for the protein quality traits, as I mentioned earlier, and then for the agronomic traits as well. So these are the beautiful pictures of our farms where we conducted our experiments. Now coming to the results, so we performed analysis of variance for all the years, and we used the 2 different models, because in the year 1 we did not have location as a factor, but in the year 2nd and 3rd we had a location. So here we used a term environment, which combines the location and year in together. So here you can see here I did not mention all the effects. I have just highlighted a few ones. So the genotypes were significant for all of the traits in all the 3 years. And also I am working on my protein quality traits yet. So here I am presenting the data for agronomic traits, which are days to flowering, days to maturity, plant height, and pod height. So this is the data for agronomic traits. So similarly, we conducted the cluster analysis for different agronomic traits, and we found different clusters for different traits in the different years. So for plant height and pod height, we wanted to increase the plant height and pod height for the mechanization of the harvesting process. So we wanted to have the genotypes which have high values, but for the days to maturity and days to flowering, since we want to fit it into a shorter duration, we wanted to have a less mean values for the genotypes. So that is why the clusters are varying in all of the years for the different traits. So by using this all cluster analysis value, we find out a few accessions which were common in different years. So for the days to flowering, we found this accession highlighted in pink font. So it was having a less number of days of flowering, days to maturity, but it has good plant height. And similarly, the different accessions colored in red, they have good plant height and pod height. And similarly, we identified the different accessions, different years. So by using these accessions, we planted a crossing block, and we make crosses. And here it is just a process that how we made crosses and how are we evaluating them. So now we are having F2s in our greenhouses, so we are advancing them. So this is just a summary or picture of my lentil experiment. So we had a lentil association mapping panel. We are following a phenotyping and genotyping pipeline for this panel. And phenotyping pipeline, we had a greenhouse experiment where we tested the protein quality traits, and the field experiment where we tested the agronomic traits and protein quality traits both. And in the genotyping pipeline, we get our DNA samples prepared, and we source them for whole genome sequencing. Then by using both phenotypic and genotypic data, we will be conducting genome-wide association study, identify the SNP. And since we are in progress for our population development, in the future, we will be making prediction models for proteins. So with that, I would like to acknowledge my advisory committee members, Dr. Dil Thavaraja, my major advisor, and other committee members. And I also want to thank my funding agencies and Clemson University for their support. And at last, the Pulse Quality and Nutritional Breeding team. Thank you so much.
So greetings to everyone. Nice to meet you all. My topic is a throughput phenomics pipeline for pulse crop nutritional breeding. And to begin with, I will start with a very short introduction to pulse crops. Basically, the pulse crops are known as, especially the pulses are known as the dry seeds extending from leguminous family crops. And they are mostly consumed as nutritious whole food And pulses are rich in both macro and micronutrients. And of the macronutrients, you can see carbohydrates, proteins, and fats, which the carbohydrates taking up mostly up to 60% of their seed matrix, and having and that in there about 20% of the carbohydrates are prebiotic in nature, which means they are low in digestibility, low digestible. And then also proteins take up to 22% in pulses, and fats nearly 5%, which fats are very minor proportion in pulses. And then minerals are a major proportion considered in micronutrients, and ferric iron, zinc, calcium, magnesium take up that proportion of minerals in pulses. And also carotenoids, which are pigments, also take up the micronutrient proportion in pulses. So when we are heading to phenotyping, high throughput phenotyping is necessary for fast and reliable analysis of these nutritional traits and also agronomic traits for breeding pulses. So current challenges. In conventional phenotyping, which is in contrast to high throughput phenotyping, has challenges related to measuring and estimating these traits, which are both the agronomic and also nutritional traits. So when we look into the nutritional traits, their analysis with conventional phenotyping, which is mostly on chromatographic methods, is labor intensive, less in throughput, and also very sample destructive, and also you use expensive instrumentation, which is mostly the related to chromatographic instruments that we use. It's very expensive. And also when we look into the agronomic traits, we have the same challenges with labor and throughput, and also at the same time there is a lot of subjective errors with traditional methods of phenotyping, which includes manual scoring methods. So these aspects create a phenomics bottleneck in conventional phenotyping, and therefore we are heading towards a high throughput platforms like constructing. But let's move forward towards phenomics bottleneck toward the nutritional traits. Actually, when you consider nutritional traits, you have different like carbohydrates, proteins, and fats, as I mentioned earlier. And you have to undergo extraction and digestion before analysis, where the analysis is being conducted with chromatographic instruments, which are even high-end, but also very expensive at the same time. So extraction and digestion steps take long time to process, even 24 hours for a batch of samples, which is like 24 to 30 samples, but still it is very labor intensive in terms of extraction and digestion, and it also so people have to it, and also very labor intensive and generates a lot of hazardous chemical waste, which is further expensive for waste disposal. So this is a kind of a phenomics bottleneck associated with nutritional trait analysis with pulse crops and also other crops associated with breeding. So the solution we brought up for this is Fourier transform with infrared spectroscopy. This is a kind of like a non-destructive phenotyping platform that we developed from our program, but it is applicable for many other crops, crops. So when we consider this, it's not like the traditional methods. You just analyze a small amount of flour from a single seed, and you can get results to multiple traits from a single scan of a small amount of flour, so that macronutrients and even the protein digestibility, nutritional quality can be assessed at the same time in a single scan. So let's dive more into this. And also it takes less than 2 minutes mostly. It's very quick. So what happens here when deep diving into this is that you place a small amount of seed and you get a mid-IR spectrum which is very simple, and you see that special bands associated with different traits of interest. So what we kind of do is we do chemometric modeling considering these regions based on partial least squares regression and develop predictive models for high throughput phenotyping the nutritional traits. So we can see that based on our results that comparison with traditional methods, our FTMIR results are comparable with the traditional or the conventional methods that we've been used, most chromatographic methods. And there are no significant differences between the FTMIR and the conventional methods, indicating the applicability of this technique for high throughput phenotyping. So now, at the current stage, we look into different chemometric models with FTMIR, and now we are at the stage of combining all the models into one platform, which we consider it to be a Nutriphenomics measure what you eat in less than a minute. It's a web application which we have kind of developed at this time based on the Fourier transform with infrared spectroscopy, so that it is intended to be deployed to the growers and breeders in South Carolina by the mid of summer, and also to nationwide breeders for application of chemometric analysis or high throughput phenotyping for these nutritional traits. So right now, for growers and breeders and even for the industry, what the users are just need to do is, like, if they have the software, they can just upload the spectral. They need to have a spectrometer, and they have to upload the spectral details in terms of text files or CSV files, and we are targeting other formats of the files, and they will get the results really quick in like less than 60 seconds. So right now, all these chemometric models are built into this platform, and it is been targeting these 3 crops at this time, and it can handle around 2,000 samples at a time. So that is very high in throughput, and this is targeting for most of the remote users to provide these services. And next, let's shift to the phenomics bottleneck associated with agronomic traits. So mostly these agronomic traits, when you look into that, they are mostly plant height, canopy height, and also days to flower, days to maturity, and includes even lodging, plant depth, and everything. So manual methods like manual scoring techniques are being commonly practiced to analyze these traits from the field, which is even labor intensive and also subjective to a lot of error. So to eliminate that, we have brought up a new platform, a drone platform, which includes deploying Skydio Extend for high throughput phenotyping. And we started this with pulse crops, and we are at the stage of extending it to other crops, which are very popular in South Carolina. So a brief pipeline for this is we fly the Skydio drone for around 20 minutes and capture 300 to 400 images of the field at a height of 25 meters. And we combine the images into a 2D map. We call it automosaic. And then we use a software, kind of, we call it QGIS software, to kind of extract and normalize green-red difference indices, and we use support vector machine, or we also use other machine learning models to fit the data for predictive analysis. So right now our models are getting advanced. This is based on the first year data, but we are at the point of expanding this analysis. So it's at the stage of construction. So, and also we've been using this drone images even for other aspects, like count down the days to maturity. So we were doing flights at 10 meters and collecting data to create 2D maps and extracting the data from the field, a random flock from the 2D map, and we just extract the white spots in the green background, which we were targeting to count the number of flowers using a computer vision approach and apply the results into the plot like this. And then later, the final goal is to apply it for the days to maturity. So this is at the stage of construction and will be coming up as a new model for measuring the days to maturity for the breeders and growers. So as a conclusion of my project, we were able to develop the FTMIR spectroscopy technique as a non-destructive high-throughput phenotyping platform to be to be used by breeders and food application scientists. And it is at the stage of development and will be released as a software package in the mid of summer for remote access. So many people from remote users can use it. And also, we have developed photogrammetric models based on unmanned aerial systems or drone images with flight times of 20 minutes to capture the images. And these models are at the stage of development, but they will be released very soon. So, and I acknowledge my advisory committee, especially my advisor, Dr. Dilthawaraja, for giving me this opportunity to work in the program, and also the funding agencies, and special thanks to all the colleagues who've been working with me on this project. Thank you so much.
Okay, great. Good morning, everybody. Excuse me. My name is Mark Dempsey. I'm a PhD student, as Dr. Dill said, and I've been working on taking the first steps toward adapting organic production in the Southeastern U.S. Excuse me. As Sonia has mentioned, lentil is a nutritious crop of global importance. It's grown across a range of geographies and production systems. It has a long history of cultivation back at least 8,000 years, originating in the Fertile Crescent area. And recently, there's been increasing interest in lentil because it's a well-balanced, nutritious food. It's high in protein, vitamins, and minerals, low in fat, rich in prebiotic carbohydrates. Within the U.S., the production of lentil is concentrated in the northern states, particularly in dryland areas, and it's grown, importantly, as a summer crop, approximately from May to August or September. However, we believe that it has potential as a winter crop in the southeastern U.S., filling a different cropping season. Yet, it hasn't been adapted to the southeastern U.S., owing mostly to its limited disease resistance. Here in the southeast, we receive a lot more rainfall than in those dryland areas of the northern U.S. And it's a relatively small statured crop without the ability to compete with some of the weeds that are present here in the southeast, which is particularly problematic for a small statured plant as it reaches the end of its growing season, begins to lose canopy as it matures, and then our early summer weeds can really take over, as you see here. However, we believe that organic grain growers in the southeastern U.S. stand to gain from lentil adoption in the region. It's an easy swap for winter wheat. It uses the same season, same equipment, planting, harvest, and in-season management. It can lengthen crop rotations, lessening disease and insect pressure over the course of that crop rotation. And it can improve nitrogen management for the subsequent crop. However, in order for that to be successfully adopted, we must identify lentils that can resist diseases and compete well with weeds, which really just means being a large statured lentil. And the lentil has an upper limit of how big that crop can get, but we want to focus on finding ones that can produce a full canopy and at the very least suppress germination or early growth of weeds before they get too big. So in order to tackle this challenge, we implemented a 3-year study in South Carolina. Sonia, who presented earlier, presented much on we shared the same project, and she's already given some other background on that project. And in order to implement this, we established 238 lentil genotypes. They came from the lentil single plant derived core collection, which is the USDA collection that's fairly diverse. You'll notice that this number is somewhat smaller than Sonia had presented because these were the sole genotypes that were present across all 4 environments. We planted this study at 4 environments. This was at 2 locations for 3 years. This was at WP Rawl Farm for 3 years. That's in Pelion, South Carolina, and it's located in the Sandhills region, which is a 95% sand soil with a really low clay content. And in 2023, we also implemented this at Clemson's Cherry Organic Farm, which is in the Piedmont of South Carolina and has a relatively high clay content at 20 to 25%. It's a sandy clay. And so we have these contrasting soils and certainly factored into disease pressure, et cetera, that we observed at those two locations. We implemented this using an alpha lattice design. That means that within each rep, we had a series of incomplete blocks. An incomplete block is simply one that consists of, that does not contain all genotypes present. This is intended to account for spatial variation within an environment and across environments. And we used CDC Maxim, a cultivar out of Canada, as check plots, which help to account for spatial variation as well. Plants were planted February 1st to February 20th, depending on the environment, on 38 centimeter or 15-inch rows, planted at 3 to 5 centimeters deep. And in the first 2 years was planted at 130 seeds per meter square, and the final year was planted at 86 seeds per meter square, in part due to seed limitations, but especially because we wanted to improve visual assessment of plants. There was no seed or fertility products applied. Again, this was an organic, both certified organic farms, so organic production systems, and weeds were controlled mechanically. So early in the season, tractor cultivation was employed, followed by a modified wheel hoe like you see here once the plants grew a little bit too big to be having a tractor run through the field, and supplemented with manual hoeing as needed. And then during the growing season, we quantified 6 agronomic traits. This began 50 days after planting and ended 130 days approximately after planting. That's days to flower, days to maturity, and then all of the other traits were quantified in between those dates. So for disease score, we quantified this on a 1 to 5 scale based on the percent of the canopy that presented visual symptoms of disease. Pretty simple measurement. For plant height, we simply measured 3 plants per plot. For canopy cover, we used digital images and a software program that measures the number of green pixels within that digital image. This is called fractional green canopy cover. Days to flower is the number of days after planting when 50% of the plants within a plot are flowering or have open flowers. And days to maturity is when 90% of pods in a plot are brown. And then we also scored lodging. Lodging, if you're not familiar with this, is when plants fall over prior to harvest, making them difficult to harvest mechanically with a combine. And so we scored this on a 0 to 5 scale based on the percentage of lodged plants. And I want to just reiterate that both the disease score and the lodging score, the lower the score, the better the outcome, and the higher the score, the worse the outcome from an agronomic perspective. For our stats, we use mixed effects ANOVA, testing the effects of genotype environment and genotype by environment. And then we used block nested within rep, nested within environment as random effects. We then calculated estimated marginal means to account for spatial variation, and these EM means were used in all downstream analyses for post hoc testing. And because this data set presented a large, complex, multivariate data set, we used hierarchical cluster analysis to separate genotypes into groups. And for any of the listeners or viewers who might not be familiar with this analysis, it's a quick and somewhat clever way to cut through a complex data set using patterns within the data that can find consistent patterns across both traits and environments. And we also, just to try to dig in a little bit better into the data, created some threshold screening values to manually screen the genotypes using the EM means, where we said, what if disease score less than 3 is a reasonable threshold where we can capture a good number of genotypes and have a plant height greater than 25 centimeters or about 10 inches, and have a canopy greater than 15%, and a relatively low lodging score. So this manual screening set up based on these threshold values is intended to narrow in on any otherwise favorable genotypes found through the cluster analysis. And we'll get to that in the results. Okay, so for results, we're going to focus on the cluster analysis results because it's a very high-level summary that we think is very useful for narrowing in on those goals of identifying genotypes that meet those 4 to 6 agronomic trait criteria. So to begin, this graph represents those the results of the analysis, which separated genotypes into 12 groups or 12 clusters. And this focusing on disease score here on the y-axis. And you can see that each dot represents one genotype within that 238 genotype population. And here I've got a red box around 29 genotypes that presented relatively favorable traits of having low disease score and high plant height, canopy, and low lodging. So you can see from this graph that the clusters 7 and 8 are relatively low in disease score, but have the box around them because they're favorable for other traits as well. And you'll notice that some of the genotypes within clusters 7 and 8 have a blue outline on them, and that's to indicate genotypes identified by that manual screening process using the threshold values I mentioned before. Now, again, the genotypes in clusters 7 and 8 are somewhat unique because in addition to having low disease score, they're also relatively tall and have high canopy cover. So these are our 3 core traits that we believe are going to be most favorable for adapting lentil to the Southeast. Days to flower and days to maturity are important simply because we want to get a handle on when this plant's coming out of the field. It's reasonable to expect that the quicker it comes out of the field, it's going to be exposed to less disease and weed pressure, and so faster is better. But also, and perhaps more importantly, because we're here in the Southeast where summer can come on quickly and get pretty warm fast, avoiding that early summer heat is going to be important for ensuring good yield and nutritional quality of those lentils. And you can see here in the graph that days to maturity has actually a fairly narrow range among the genotypes in clusters 7 and 8, indicating that we might not have actually very much wiggle room for breeding early maturity, but instead we might have to rely on agronomic practices, perhaps like an earlier planting date to achieve an earlier harvest. So 120 days, give or take, is what we saw for these 2 clusters. And that puts us at early June harvest because we planted in early February. And if we wanted to avoid that early June heat, then perhaps we're going to have to just push back planting date. And finally, for lodging score, most lentils scored pretty low for lodging, below 2. There was only one cluster that particularly scored high for that. And so it was more or less within group 7 and 8 that we found that full combination of low disease, high plant and canopy cover, or sorry, high height and plant canopy cover, and that low lodging. Again, and if you track those blue outlines, you can see how the 10 best performers for those traits that met the threshold criteria performed across these 6 traits. So in summary, we identified 29 genotypes with those favorable traits. The manual screening using threshold values identified 10 within those 29, and those are shown here. In the interest of good science communication, I wanted to make sure we put those genotypes up on the screen, and the 10 are bold-faced. For conclusions and future work, again, those genotypes in clusters 7 and 8 may have greater disease resistance pending further research. They likely have greater ability to compete with weeds owing to their large stature and, of course, scored relatively low for lodging. The manually screened genotypes that meet those threshold values or criteria are likely good candidates for future breeding. Importantly, though, I certainly want to flag that having a better understanding of the pathogen community affecting lentils in the region is going to be important, and understanding disease identity on the ones that we observed in the field, and then culturing those for a more careful approach of screening for disease resistance is going to be an essential step for understanding actual disease resistance among the genotypes that we've tested. And so what we have done here is a large snapshot that's relatively coarsely grained for disease susceptibility, and it's only a score of disease incidence and not actual resistance. And so this is a first step toward understanding potential resistance among genotypes that were screened. And overall conclusion, this is a first step toward identifying parents for an organic lentil breeding pipeline for the region, which can help increase both regional and global lentil production, contributing to food security and nutrition. I'd like to thank my advisor, Dr. Dill, my committee members, our lab team, and our funding sources. Thank you.
Thank you so much, Mark. Wonderful. So since we have only 10 minutes, I'm trying to get one question at least for everyone. So let's start with Mark. Mark, you have identified wonderful 29 genotypes, which we incorporated already in the greenhouse to our crosses, and we are making populations out of that to go out in the field. But what do you think of when our growers can actually adopt these? Because you have 10 years of experience of organic agriculture. What do you see the constraints of the adapting agriculture in lentils into the organic agriculture in South Carolina?
So the question is primary constraints to adoption or production?
Exactly.
I believe it's still understanding diseases, especially if grown in heavier soils. So in the sandhill regions where we've got 95% sand, it's a lightly textured, well-drained soil. I don't expect, and our experience would tell us that disease pressure was low. And if there are growers in the sandhills that are interested in lentil production, I think this crop is almost ready to go. There are cultivars that we observed that performed really well. They were big and bushy with low disease incidence or disease scores. In the upstate region where the clay content's high and soils are basically wetter, I think we have a little way to go. And I think we could use at least another year or two of screening to have a better handle on disease dynamics. And of course, then that needs to be taken, I believe, to the greenhouse for a more controlled environment to understand actual disease resistance among those genotypes.
Absolutely. So that's kind of a pipeline that we have right now. We have some F2s. It's also constraint is to make the crosses, right? It's that time.
Yes.
Yes.
Yes. Yeah. For anything beyond an existing cultivar or a genotype, if we want to improve upon it, that has become a constraint for us as well.
Yeah.
Yeah.
And exactly. So one other question coming out. This is for Mark. Alex, one of our participants, want to know, I would like to know if a PCA could be used instead of a cluster analysis. Would you use the PCA instead of cluster analysis? What is your hypothesis? So why did you do that?
I preferred, in this case, the PCA because it was a simple way to just pull groups of genotypes apart. It's a simpler analysis that tests, instead of for correlations like in a PCA, it calculates dissimilarity among those genotypes. And so I found it an easier way and perhaps more intuitive way to just group genotypes instead of creating what I call a synthetic variable of a PC, which just makes it it's a bit of an abstraction. So I just prefer it because it's an intuitive analysis compared to PCA. Thank
you. Yeah, that's kind of a comprehensive question, right? I have two more questions for you, Mark, before I move into other two students. The other question is from Dr. Branham. She's our vegetable breeder. She wants to know, it's known what diseases are most common for South Carolina for lentils. She wants to know most common diseases for South Carolina. And where would you be ID any of these in your field? Would you be able to? I think we did, right?
Yes, we did. Yeah, and that's some results that I didn't go to in any detail. The two most common ones, at least that we were able to identify down to the genus level, is Pythium as contributing to root rot, as well as Fusarium contributing to root rot. And we did observe some Fusarium wilt, but it was really small compared to Fusarium that was associated with root rot. It was Fusarium oxysporum once or twice, and then we also observed one or two other species of Fusarium. These were all just associated with the root rot, and much like with the Pythium as well, we need to culture and reinfect to confirm that those are the diseases contributing. I will also mention that Pythium is often more of an early season oomycete infection that affects it before it gets too big, and we observed it a little bit later in the season, and suggesting that Pythium might have been there, but might not have been the main downfall for the oomycete disease that we observed. And there could be another unidentified oomycete out there, like a Phanomyces, which is suspect but unclear because it's a very difficult one to culture. We did test these, get them diagnosed at two different labs, one at Clemson, one at Montana State, where they handle a lot of pulse diseases. And the oomycete was inconclusive at Montana State.
Absolutely. And for the one more question from Sandra, I can answer that. Root rots were the most problematic. Yes, it is. So I have F8, F9s to be released, and we see some of them are actually affected by black root rot, which is identified by the pathogen from the ID lab for the peas, and Mark was working on lentils. So yeah, it's kind of a challenge, and I'm not trained for diseases. So I might come to you, Sandra. So one last question, because diseases are very important, and we have a couple of pathologies in this group, and asking for Mark, how are you distinguish between whether these pathogens originated from different environmental sources? I think you did that, where there's pathogens coming from different sources, right?
So if the question is whether it came in on our seed or whether it was native, we don't have a good control or a good handle on that. We expect that we grew this from seed from USDA and Arcarta, and hope that we didn't, in our greenhouse grow-outs, ever add any pathogens. I suspect that pathogens are native to the fields here and not necessarily introduced. That's my gut feeling. We don't have any good information on whether that's the case.
Yeah, that's absolutely correct. And thank you so much, Mark. It was wonderful. So I'm going to move to Amod. I think you guys all can turn on your camera, so then so that way that we could switch if someone wanted to answer. Amod, so like your technology with the high throughput phenotyping, can you mention about the cost, about using the general lab mention, like let's say the protein?
Yes.
We measure protein quality, amino acid. My lab, I am good for the nutritional traits, which we have a beautiful phenotyping lab. So can you little talk about the cost of our new technology that you developed, as well as what our lab does?
Yeah. So based on the lab part, the conventional part, the cost of analysis is very high for protein and for analyzing all the carbohydrates and proteins. It costs like $200 to $300 per sample if you have to do amino acids, like a profile of amino acids, which is very expensive. And also for carbohydrates and other traits, which relates to wet chemical analysis, it is very expensive. So, but for this approach, we can analyze all the traits, carbohydrates, proteins, fats, and everything, for like up to a cost of like $10, which is including everything at the same time. But for individual trait with the conventional methods, it's expensive. So the novelty is that we cut down the cost to like $10, which is very cheap and reliable, so that many breeders can use this technology. So
absolutely. And again, there's a question from Dr. Banham. She's asking, have you assessed the correlation between flower number and the yield? Not yet, right?
Not yet, but I'm working on that project. So I have to work on improving that flower counting capability of computer vision. And I'm at the stage of working on that. So, yeah.
Yeah, that's kind of another month of work. And then you probably will have it, because we have 4 more weeks to harvest, and Tristan is preparing for harvest.
Yes.
We'll have the data. All right. I think that's the questions for high throughput. Do I see anything else coming? All right. Okay, rest of the questions for Sonia. All right, Sonia, you did a wonderful job. Thank you so much. And there's many questions. So let's start with one of our bioinformaticians' questions from Carolina. Dr. Carolina's question. After doing GWAS analysis for fine markers linked to a trait, what are the next steps in terms of breeding? Haha. What can be done with the information? It's a few multiple markers. She's asking you all the defense questions. So what do you think?
Yeah, after having the SNPs, we can see how heritable individual SNPs are and how is the total heritability for the different SNPs. Based on that, the most significant markers, we can choose them to design our competitive allele specific markers, to develop our own markers, and then we can validate it on two extreme sets, which are like high protein and low protein ones. And we can see marker validation. After doing that marker validation, we can use those markers on our breeding population, advanced ones like F8 or F9. So that's the one thing we can do with them. And also so this information will be helpful to cut down the selection number from a very broad number to a small number. So this way we can make sure that, okay, we are not looking for the genotypes which are expressing it only phenotypically, but they do not have a genetic basis, but we will look for both genetic basis and phenotypic basis as well.
Absolutely. Thank you. And yeah, this all worked. So grad students are amazing to have in a breeding program with wonderful, these kind of awesome grad students. So they help my breeding program running so fast. So one of the questions, Sonia, this is you good at it. What method will be used for genotyping the panel? We had a lot of help from Dr. Boatwright. So you want to talk about how did we do the panel and the genotyping?
Yeah, so we chose the panel. Basically, we took the information from ICARDA. The most of the panel was from ICARDA and USDA. It was not only based on the origin, but it also included information like where it is coming from, the center of diversity, and where it is mostly being cultivated, like it's being mostly cultivated in Asia and most being used in that area. So that's we try to inculcate in our panel to account for diversity as much as we can. And we sequenced it by using whole genome sequencing, which I feel is a good way to cover the most of the part of genome rather than doing GBS. So it is a self-pollinated crop. It could benefit from GBS as well, but I think whole genome sequencing is still have a more plus points compared to GBS.
Yeah, so it was a 10x coverage that we did
Yes,
for
it was 10x
each individual, and we sent it out to Florida. We didn't do anything. We just grow it, send it.
Yeah, we sourced it.
Yeah, and it was like very fast. We got our genotyping data, I don't know, less than 2 months. So it was really fast. Yeah, and also we have the computing power here at the Clemson University, so we can do quite a bit of interesting work here. So that answered. And one last question, because it's 10:59. It goes to Sonia. Sonia answered most of the questions. And one of the questions were asking about how the amino acids were measured. Sonia, you want to take that?
We chose sulfur-containing amino acids for the reason because they have a distinct functional group in them, which could be identified by using FTIR, the technology that Amod introduced to all of them. So we used FTIR to measure these, all three traits: protein quality, all three traits, protein concentration, sulfur-containing amino acids, and digestibility. We used FTIR models.
Right. So actually, I had another previous graduate student, Dr. Nathan Johnson. He developed the amino acid assay in the lab because the previous methods were not correct. In his thesis, he developed the proper method. So we measured all the amino acids. And then part of his thesis send it out, the amino acid, sulfur-containing amino acid into the FTIR, and then Amod came and created. So now we don't use the HPLC, we use the FTMIR to do that. So it's 11:01, and thank you so much, all of you. And I got lots of good comments for all of you who are doing wonderful jobs, and you guys are early carriers, going to be great breeders and scientists, and I'm very proud. And thank you so much for joining today and sharing your science with our group. And this is our last webinar, and this concludes our webinar, and we will meet again in the fall. Thank you all joining today, and have a very best of summer. Goodbye.
Thank you so much. Bye-bye.